Homeostasis, Maintaining H2O Balance by Anne Howley, Chris McCurk, Tori Cooper, and Bridget Marshall. Slide number 1.
What is water regulation? Water regulation is based largely on shuffling solutes in H2O between internal and external fluids.
Excretion of nitrogenous waste through land-dwelling organisms. Organisms must rid themselves of toxic and nitrogenous waste that results from protein catabolism. Here in this diagram, you can kind of see how nitrogenous waste is excreted through different organisms and different mammals. However, land-dwelling organisms excrete nitrogenous waste differently than water-dwelling because in land-dwelling, most organisms, they're forced to store nitrogenous waste for long periods of time before they're able to excrete. And because ammonium is very toxic, they must store this for a while, and because of that, it has to be stored in the liver and converted to the urea because this is less toxic and water soluble, and so it's safer to store for a long period of time by storing toxins.
Excretion of nitrogenous waste in water dwellings. So in comparison to land dwellings, water dwellings excrete their nitrogenous waste a little bit quicker, so they don't need to store the ammonia for as long in their body, and therefore it's not toxic, so they don't need to store it or convert it to anything more safe. And they excrete this through diffusion, through skin, gills, things like that. Some of their most significant waste products are the nitrogenous breakdown of proteins and nucleic acids, and they need lots of access to water because ammonia can only be tolerated at low concentrations. So these water animals are often in the presence of water. Ammonia, like we said, ammonia excretion is most common in aquatic species, and the highly soluble ammonia molecules can easily pass through membranes and are readily lost by diffusion to the surrounding water that they live in.
Osmoregulation, water-dwelling organisms, fresh water. The purpose of this is to maintain electrolytes while getting rid of H2O.
Osmoregulation, land-dwelling organisms, saltwater. Purpose for osmoregulation is to get rid of sodium chloride within organisms while maintaining H2O. Osmoregulators are organisms that drink salt water and actively transport sodium chloride out through the gills. Examples of these are fish. Osmoconformers are organisms that maintain a significantly higher level of urea in the blood, and osmolarity is somewhat similar to the surrounding water, and because of this, organisms like sharks lose little H2O to their environment.
Excretory processes. So these processes rely on transport epithelial, which are one or more layers of epithelial cells. And the first step of these processes is filtration, which is where an excretory tubule collects a filtrate from blood, water, and solutes are then forced by blood pressure across selectively permeable membranes. And in the second step, called reabsorption, transport epithelium reclaims the important substances from the filtrate, and then they are returned to body fluid, and these can be nutrients or proteins that the body needs. For the next step, it's called secretion, and this is when toxins and excess ions are extracted from body fluids and added to contents of excretory tubules. And the last step is excretion, and this is when the altered filtration, known as urine, leaves the entire system and body.
This is the diagram of the kidney. This is important because this is where the nephrons are stored, and they're a key factor in the filtration process.
As mentioned in the previous slide, these are the nephron types, and again, these are important to know because they are key factors in filtration processes.
This is just a close-up diagram of the nephrons, and it just shows the organization and structure.
The first step of the nephron filtration steps is in the proximal tubule. This is where the reabsorption of ions, water, and valuable nutrients happens. And then the NaCl then enters the transport epithelium by through facilitated diffusion. And then Na plus is actively transported to the interstitial fluid. And then this positive charge of the Na plus transfer to the interstitial fluid drives the passive transport of Cl minus. As the salt moves, the water moves as well through osmosis, and the water and salt then diffuse into the peritubular capillaries, while other essential substances are actively and passively transported there to the peritubular capillaries. And then finally, toxic materials are actively secreted into the filtrate.
This is the second step of the nephron filtration, the descending limb of Loop of Henle. Reabsorption of water continues as the Loop of Henle is formed. Water channels formed by aquaporins make the transport epithelium freely permeable to water. No channels for salt and other small solutes, very little permeability. The interstitial fluid of tubule must be hypersomatic to the filtrate for water to move out of the tubule. Then the filtrate loses water and increases in solute concentration as it travels down the descending limb.
Nephron filtration step 3. Thin segment of ascending limb. First, filtrate reaches tip of loop and returns to cortex. The epithelial membrane that faces filtrate is impermeable to water. The ascending loop has two specialized regions, the first region being thin segment, where NaCl diffuses out of the permeable tubule into interstitial fluid, and the second region, thick segment, where movement of NaCl continues. Epithelium actively transports NaCl to interstitial fluid. The loss of salt, it loses lots of salt and not much water. Or in other words, filtrate becomes more dilute as it moves up the cortex.
The fourth step of the nephron filtration is the distal tubule, and the distal tubule plays a key role in regulating potassium and sodium chloride concentrations of body fluid. This regulation involves a variety of amounts of potassium that is then secreted into the filtrate as the amounts of sodium chloride are reabsorbed from the filtrate. And finally, the distal tubule contributes to pH regulation from the controlled secretion of H+ and the reabsorption of bicarbonate.
This is the fifth step of the nephron filtration. This is the collecting duct. So the first part is that it carries the filtrate through the medulla to the renal pelvis. The final processing of the filtrate by transport epithelium forms the urine. Approximately 1,600 liters of blood flows through a pair of kidneys each day, but leaves only about 1.5 liters of urine to be transported to the bladder. As filtrate travels along the transport epithelium, regulation of the permeability determines the extent to which urine becomes concentrated. Aquaporin channels in collecting duct allow water to cross the epithelium in order to conserve water. The net result is the urine is hyperosmotic to the body fluids, and the state of collecting duct epithelium is controlled by hormones. Concentration. Here is a diagram of urine concentration and the different steps within it. Tests for urine concentrations are important and performed to determine the ability of the kidneys to excrete H2O. Urine is produced in the kidneys. When you take in fluid or food, it passes from your digestive system into your circulatory system and into your kidneys, where it is then filtered. The kidneys then get rid of the waste products and extra fluid through the urine. Darker urine is most concentrated with solutes, and a lighter color urine is less concentrated with solutes and more water. When it's darker, it typically happens when dehydrated because there's not enough water. Homeostatic regulation of the kidney. Here is a diagram of it. The antidiuretic hormone, also known as the vasopressin, is the key hormone in the homeostatic regulation of the kidney and fluid regulation. Osmoreceptor cells in the hypothalamus monitor osmolality of blood and regulate the release of ADH from pituitary. ADH binds to the receptor molecules from epithelial cells in collecting duct and to temporary increase in aquaporin proteins in the plasma membrane. Aquaporins then form water channels and create the net effect of an increased permeability of the epithelium to water. The last slide is the homeostatic regulation of the kidney through blood volume and blood pressure regulation, and here's a diagram showing it. The renin-angiotensin-aldosterone system, known as the RAAS, operates as feedback circuit. It involves the juxtaglomerular glomerular apparatus, the JGA, which is specialized tissue that supplies blood to the glomerulus. When blood pressure of volume drops through an example of its dehydration, the JGA releases the enzyme renin. Renin creates peptide called angiotensin II, and this triggers vasoconstriction and increasing the blood pressure and decreasing the blood flow to capillaries in the kidney, and this causes the release of aldosterone. Aldosterone and angiotensin cause rise in blood pressure and blood volume. The last thing to know is when electrolyte levels are high, the body retains more water, which in turn increases the volume of the blood. More blood volume results in higher blood pressure, thus the kidneys maintain blood pressure by indirectly controlling the amount of blood in the body.